Hey everybody, welcome to lesson 10. Today we're going to be going over microbial genetics. So this is pretty much just going through the basics of genetics and how that impacts microbes and what that really means. It's a topic we've somewhat visited in pretty much all of the past lessons. We've talked about it a little bit, but we're going to be doing a little bit more of a deep dive. Now, we've talked about DNA viruses versus RNA viruses, and we're going to go into that more in depth as our first step because these are the building blocks of all of the proteins that end up making, whether it's a virus, a bacteria, or a whole human, they're the building blocks that end up making the proteins that compose the organism. So DNA is deoxyribonucleic acid, so that's why it's called DNA, deoxyribose, and then N for nucleic, and then A for acid. And this is a sugar. So that's getting a little bit too much into the weeds, in my opinion. You should know it's a sugar. You should know that it is a double-stranded helix, so it weaves into a helix. I'm sure you've seen that in previous courses. And it also has four bases. That's A, T, C, and G. And those are going to be complementary. So A to T and C to G are going to go together. And then when we go to RNA, this is just ribonucleic acid. It is also a sugar, ribose, and it also has four bases. But there's one difference, and that one difference is the T. The T in RNA is instead a U. So A to T, A to U in RNA, and then G to C is always G to C. You've probably heard a lot about RNA. So there's three typical types. There's mRNA, tRNA, and rRNA. mRNA is the one that everyone knows about because of the mRNA vaccine. This is when the mRNA is being used. It is being used as a messenger RNA, the M, and then it's going to be directly transcribed. So you're going to take that mRNA, it's going to turn into DNA, and that's going to turn into protein. And that's generally how a lot of viruses that are RNA-based viruses are going to work as well. They're going to have these mRNA components that get transcribed into ultimately proteins. Now, we can think of it in us as people as DNA being permanent storage of information that encodes proteins, and RNA being the copies that are taken out of storage to produce the protein. So you have your nucleus, and in that's all the DNA and the chromosomes. And what happens is you have RNA that gets coded for and then sent out to be turned into your proteins at the end of the day. Um, so a picture of this is very, very helpful. It's kind of hard to describe in the 3D space of a cell and everything going on, but if you look at a picture, and there's some great videos online, it'll become pretty apparent how that whole system functions. You can get it into a bunch of incredible detail with it as for everything else, but that's the general workflow. So you can take DNA to RNA, which is then going to be used for protein synthesis, and then you can have, uh, depending on the virus, you can have some wacky stuff happen. So DNA, RNA, protein. That's all you got to really remember in that workflow is you're going DNA to RNA, then to proteins. Now, this is super helpful to us in general because everything has a certain amount of DNA that is conserved as it passes on its lineages. Of course, there's mutations in the form of, mammals and most higher order life were a two-part pair. So there's a mother and a father, and then that makes the babies. So you're getting two halves that make a new whole. And through all of that, we can either track the lineage of a bacteria or a virus or a person to these different webs of origins. And that is not only useful in identifying, a long lost uncle or something like that, but also for identifying specific microorganisms. If someone's infected with a virus or a bacteria, you can take their blood and then you can do a PCR and check for that genomic information, that DNA or RNA information that then gives you your identification of that pathogen, right? So that is a huge benefit for us in healthcare. It's also going to be indicative of a phenotype. You can look at the DNA or RNA and you can extrapolate from that these what would be observable traits. So the proteins that are being produced, what enzymes are being used in the metabolism. We visited metabolism recently. So what enzymes are actually being used in there? What different variable structures are being made. So does this have a flagella? Does it have a messed up weird flagella? Is it a new form of a mutation that makes this flagella better? We can identify all that from looking at either the DNA or the RNA. And this can also be a big deal environmentally, in environmental sciences. So if you're at a lake and there's a lot of weird algae, it's been upset, the fish keep dying, you could just take a sample from that lake, run a wide spectrum analysis, and figure out, okay, we have these specific bacteria or algae in the lake that are throwing it into this disordered state. So we need to do some sort of treatment or we need to introduce XYZ different bacteria into the system and that'll help clean it up. Right? So there's a ton of different interesting applications of being able to look at DNA and RNA. Another example, of course, is if you have a person with a genetic disorder. We only know it's a genetic disorder and how that disorder works from looking at them, doing a genetic analysis, sequencing their DNA and saying they have a mutation or they have a deletion, whatever it is, they have that genetic disorder. We might know phenotypically already, but that shows exactly what the core issue is for someone. And now, luckily, with CRISPR-Cas9 and a lot of different scientific advances, we have genetic treatments that are kind of coming out pretty much every year. There's a new one that hits it big, and those can be used to treat those very rare conditions. So, DNA is always going to be replicated in mammalian cells. And it goes beyond that, but of course, there's some weird caveats, so I don't want to say all cells, but pretty much all cells. When DNA is replicated, there's some key different things going on. So, first, it's semi-conserved. If you have one mother molecule, you're going to get one daughter molecule of DNA as that split occurs. And you're going to have a lot of different enzymes, especially involved in this. So, there's these big supercoils that are in your nucleus. So when you replicate a cell, the nucleus comes apart, and then you have all of these supercoils that are wrapped up in histones of DNA. So think about it, you take some yarn and you knot it up about a bunch of, sticky bits of gum. That's hard to parse out. Well, you need something to parse that out, and that's going to be our topoisomerase and our DNA gyrase. They're going to relax those supercoils, pull them out, and then there's something called helicase, which is actually going to take those strands of DNA, remember they're helical, and it's going to unwind them. Then DNA polymerase comes in, synthesizes new strands, and primase lays a RNA primer onto that point, and then DNA ligase is going to come and repair that singular strand. So the steps here are you relax the supercoils, you unwind the strands, DNA polymerase comes in, synthesizes a brand new strand. So you went from splitting two strands to now you have an extra pair. So you have four. And on that new strand that's made, a little bit of RNA comes and pops on, and then you have something else come in, and that is going to be your ligase, and that is going to take new DNA, A, T, C's, and G's and put it onto that DNA strand. And that's how you get your copies. So that's what happens in most eukaryotes. In prokaryotes, they have circular DNA in a lot of cases. So it's just a circle of DNA. Of course, there's still two, but it's two in a circle. And then it's going to split and it's going to have a replication of origin. point where you actually fit in it. It kind of unwinds and winds back together as you're going around the circle. And then you just get this strand that spins off. And then it can pair up to another strand that's done with the other complementary side of the DNA. Again, this is something that's a little complicated to hear about and not see. So I think it would benefit you a lot to go just search up DNA replication for prokaryotes, DNA replication for eukaryotes. Find a quick, 2-minute video on YouTube, and I'll see if I can find some of those as well and provide links. Then we get into transcription translation. So transcription is DNA to mRNA. And I apologize. I've probably mixed these up. I've always been very, very bad at it. I've been reprimanded on many a test for flipping these two. I'm dyslexic, so it's pretty much impossible unless I'm staring at them to remember which is which. But transcription is DNA to mRNA. The mRNA is what is going to then go in and create protein. So you have transcription, which is DNA to mRNA, and then mRNA to protein. And that's why the mRNA vaccines work so well, because you get the shot, the mRNA is already there, it just starts making protein, and that's how those work. So your mRNA, it's going to pair up with some ribosomes, it's going to be read in triplets. You're going to have something called tRNA, which comes in and has 3 amino acid pairs that correspond to the mRNA. And it's going to read those and it's going to be like, okay, awesome. I know what this is. And then you have rRNA, which actually forms that full ribosomal structure. So you have a lot of different RNA doing a lot of different jobs, but at the end of the day, it is the mRNA directly encoding the proteins that you're getting out the other end. And the other two forms are just helpers in that situation. Why that's important to talk about is because this is where a lot of your downstream mutations are going to actually occur is on this translation. And that's a good thing that sometimes this is read wrong because that means that if you have a substitution, things like that, with the tRNAs, the three bases, sometimes that last base, it's called a wobble base, pairs up differently. And you can get some slight variation in proteins made, but we have machinery to take care of that. That can get degraded. So this translation is not 100%. Nothing in biology is 100%, but it has downstream processes that will take a messed up protein, degrade it down, get rid of it, recycle it, and solve these issues. The real problem with mutations is at that higher order, that transcription side in the DNA. Because if your DNA is messed up, then everything that comes after is also going to be messed up. And that's why everyone who has a severe genetic condition, they're going to get treated with gene therapy because there's really nothing else that you can do. You can have supportive therapies, but they're going to need a gene therapy to solve the issue with their DNA from making, whether it's aberrant proteins or there's, just something wrong in the coding. So something's going wrong that is a bad enough case to be a disease. In most people, normally, this is actually just going to turn into cancer. We don't have a large scale issue with genetic disease because the issues have to sort of be baked into the DNA. When your DNA in a part of your body gets damaged, that's in a specific cell, and we've already grown. We're adults, so we're not coming from one cellular source. So any issues we have needed to come from that source. If the issues come later, they're happening in singular cells, and they can cause issues, but that issue is normally cancer. So that's a long way of saying that if you have a genetic disease that usually was in place at the time of your first division from, egg and sperm coming together and getting those first divisions, it occurred somewhere in there, or it was passed down. And that's why genetic diseases are generally passed down from person to person. There's a couple that happen randomly, but they're usually passed down. And on the other side, if you get damage to your DNA, it's typically going to form aberrant things that your body takes care of, or it's bad enough that it turns into cancer because everything's gunked up. That cell doesn't work, it starts freaking out, has a bunch of problems. Now, there are many, though, mutation types. Silent is there's no change in proteins. And usually we measure this by there's going to be no change in the actual protein at the end of the day, or there's going to be a major change in the protein at the end of the day because the protein's what we're actually having interact with other things in our body. So silent is no change to the protein. Missense means that there is a single amino acid change. This sometimes doesn't matter because you have these tRNAs with the three bases, and that last base, that wobble base, sometimes is going to be replaceable by another tRNA that encodes for the exact same thing. So you end up in this condition where you could have a mutation, but it doesn't change function. It might, but not always. There's what we call nonsense mutations. This means you get a stop codon. Of course, there's a code in there that stops the reading of the DNA and says, hey, this is where I end. This is the end of the protein. Down here is more protein, but stop here if you're making this protein. And if you get a premature one, it truncates the protein. And that can mean that the protein is non-functional, or it can mean that the protein works absolutely fine. And for whatever reason, evolutionarily, we have a little bit of extra end on that protein that might make it work better. But if it's cut off, it doesn't stop it from working completely. Then there's frame shifts. These are the very, very bad ones. So this is when you get a complete deletion or an insertion, and that shifts the entire thing you're reading. So if you think about DNA is you're looking at letters in a string that's endless. If you just take that whole thing and shift it down, and your start point's the same, and your end point is the same, that is going to make something that you do not want to make. And these frame shift mutations you do not want. They're going to be very, very bad. They're almost universally going to be non-functional proteins, sometimes very, very bad for you, non-functional proteins. These are things that end up being your protein that gunks up the cell and leads it to be cancerous, or your protein that gunks up the cell and leads to immediate apoptosis. And if that happens somewhere like your neurons, that can be bad. And we get cascade events in these kind of mutations sometimes similar to prions. So prions can be made from this kind of mutation. It can also be made from other kinds of mutations, but you could get a frame shift that makes a prion, and then you're going to be developing that continuously, and it's going to keep gunking things up and be a really, really big problem. So frame shifts are really, really bad for us. And this, especially the frame shifts, are typically what you're going to see leading to cancer. And the reason for that is frame shifts happen often when you have two strands and they get broken. And then these ends degrade and these ends degrade, and your body just it tries to repair it, but it doesn't know what's there. It has no way of knowing exactly what's in that space after it's broken on both sides. And that's why we have the pairs, because if we had one, it would break all the time and you'd never know. So DNA is paired up with complementary bases put together, and when that has a double-stranded break is what we call it. When your body puts it back together, it's usually either missing stuff or it's adding stuff in. So you get these frame shifts. And that is what is traced over and over again as really leading to cancer. Of course, there's all the environmental things. If you smoke, if you drink, and it leads to cancer too. And that's because when your cells are under pressure, there's more things that cause these breaks and cause them to be less efficient at fixing these breaks. So that is one of the theories as to why we get cancer in the first place, is from these double-stranded DNA breaks. Now, bacteria are a lot different. So, like I mentioned, they have these helical DNAs, and you're going to create new circles of DNA off those. So they're just going to replicate that way. But how do they give each other their information? That's a really hard question to answer when you're just thinking through how do we as organisms give our information, and that's through two people pairing up. But bacteria just divide. So how do they do it? The way they do it is through a couple different weird avenues. One of them is transformation. This is uptake of what is sometimes termed naked DNA from the environment. So if there's just DNA hanging out, sometimes it'll come in and be taken up by the bacteria, and the bacteria will say, "That's cool," and integrate it. More likely than it just coming in that way is it's coming from viruses. So viruses infect the bacteria has ways to kill off the viruses, but there's extra DNA floating around and just kind of gets integrated in the same way we integrate DNA from retroviruses, for example. Then there is transduction. So this is DNA transferred specifically by bacteriophage viruses. This is very different than transformation because there's a lot of machinery going on into that to integrate the DNA into the bacteria. This is not a straightforward, oh, it's just going to dump DNA in and that's going to be taken up through transformation. No, transduction, there's machinery that's transduction machinery that is going to get in there, and then it is going to overtake and put new DNA into that circular DNA from the bacteria. There's another thing called conjugation. Some bacteria do this, some don't. And this is direct transfer of genetic information. The bacteria essentially forms a tube, stabs another bacteria, and injects DNA into it. Kind of gruesome when you think about it too much and put it into a human-centric viewpoint, but that's one of the other ways that they transfer their genetic information to one another. And this is how antibiotic resistance spreads. Usually they're giving each other good DNA, so they're shooting off a bunch of antibiotic resistance at each other, and then they're all antibiotic resistant. It's bad for us, it's good for them. So that's why they're going to do that. And these all create what we call recombinant bacteria. That's typically used in a research setting. So recombinant bacteria is something that we as researchers have modified in the bacteria to make them do something for us. But it also applies in this case where they're either going through transformation through random uptake of DNA, transduction, where you have your DNA transferred by bacteriophages, and those are bacteria-specific viruses, or this conjugation where you're having the bacteria transfer DNA to one another. Let's think about this a second. So, I want to touch on something very quickly, which is that your genes are regulated. And this again kind of goes back into that higher order mammalian space. You have different ways of regulating your genes, and these are things called promoters and operators. There's also uh, whether you have the spools of DNA tightly wrapped and not accessible, or partially unwrapped and somewhat accessible, or very unwrapped and very accessible, while you are just having normal cellular life cycle. And this is not when it's dividing and making more DNA. This is just when it's all doing its normal day-to-day cellular job. And there are these different ways of regulating, and we as humans, and this applies to lizards and a dog and an elephant. So as higher order animals, multicellular animals, we have a lot of ways of doing gene regulation, and that's very important because we have very different cells, whether it's our skin cells, our muscle cells, the cells in your eyes, the cones and rods at the back of your eyes, your neural cells. There's a bunch of different cell types. And that all occurs through some sort of regulation. It has to, because we're only still coming from realistically one DNA total set. So what gets produced when and where and how is a huge deal. And that's controlled with all these different regulators. And you're going to learn these a little bit more in depth, but that's why they're important, and that's why we study them in depth. That's why we know a lot about them, and why we are interested in targeting different regulation patterns for things like anti-aging therapies or various treatments. There's a lot of different ways we can look at that and try and control that are beneficial. And when we take a step down and go back to the bacterial world, they also have some of this, but it's usually in this operon class. So operons are essentially just going to turn your genes on and off. They're not going to regulate in a detailed way. It's just on, off, on, off. That's it. So you have your promoter, which is required for all the machinery to come and transcribe your DNA. Sorry, yeah, transcribe. That's right. I checked myself. So that's all required for the transcription of your DNA. And then you're going to have operons, and operons can sort of block whether you're going to have that promoter turn on or off. So there's something that is around the promoter. So if you think of promoter as a set of DNA, here's your promoter. And around that on either side, you could have an operon. And that operon says, I'm off. So actually, you can't do anything. And the way they do that is they actually prevent binding of the machinery. So something sits on that operon and blocks the other machinery from coming and getting onto that promoter. So the operon sits here and it's got a protein on it. It's just like, okay, I'm trying to get everything going for this protein. I just can't because there's something blocking that protein DNA interaction. So one of these that we use a lot is called the lac operon. It's normally off. So that means there's something bound preventing this whole transcription from happening. And then you put lactose into the nutrients that you feed these bacteria. And then this goes, oh, lactose is here. I'm going to fall off. And then I'm going to turn because the protein fell off that was blocking it. This promoter gets turned on. And what does that promoter end up producing? Of course, an enzyme to break down lactose. So it has this feedback cycle that's really, really cool that we can use to recruit the promoter and produce something that is supposed to be the enzyme that degrades lactose. But we can replace that DNA with whatever we want. We can put anything in there. So we have these really cool systems on a bacterial level of making what we want in bacteria through manipulating these operons or the promoters or other different structural genes. There's a lot here that you'll end up learning over time, especially if you get very genetics focused. And it's kind of mind-boggling how much control there is over what each cell is producing in our bodies and how much control we can have as researchers over what bacteria do for us. So that is a really cool but very complicated subset of genetics is how do we control these things? How do we look at these things? What are they actually doing on a day-to-day basis? Now, of course, viruses also have genetics. We've talked about DNA viruses, RNA viruses, and retroviruses. So just a quick review, DNA viruses, they follow that normal replication transcription model because they put the DNA into your body. That gets transcribed into mRNA. That mRNA then gets translated into protein. And then RNA viruses typically have mRNA, and then that gets translated into the protein straight away. So that's pretty cool. There's also negative strand RNA, which they have to go back and make the DNA, and then they make the mRNA, and then they produce the proteins. And there's reasons for that I'm not going to get into because it gets a little convoluted, but those also exist. But usually it's going to be mRNA to protein with RNA viruses. And then there's the retroviruses, which go RNA to DNA to insert that DNA into our cells. And then from that insertion, then they produce what they want. So that is a very, very different type of style and why retroviruses, even though they pack RNA, are split off into this different group of viruses. So viruses, of course, because they're messing with our DNA a lot of the time, especially retroviruses, are a source of mutations and they can cause cancer. HPV, human papillomavirus, worked on a vaccine for that. it's very important to be vaccinated against it because those viruses cause cancer. It's looked at as usually a sexually transmitted disease, but there's hundreds of types of HPV. A lot of them cause cancer, and you can get it from, sharing a drink with someone, that has it. So this is one that it's caused a bunch of cancers throughout time, and now the vaccine's pretty common. So we're seeing a decline in those types of cancers, and those are head, neck, throat cancers and cervical cancers primarily. But there's a couple other types that come from those as well. Another one is Epstein-Barr virus. There's not a vaccine right now for EBV, and most people who get it are totally fine, but it's also linked to being an oncogenic virus. Oncogenic meaning it causes cancer. So understanding how all this DNA and RNA works is very important to understanding how viruses affect us, how we can use bacteria, how we can use viruses. And I really do encourage looking at a couple videos of DNA and RNA transcription, or sorry, DNA transcription and RNA translation into proteins. So if you just type in, you know, uh, DNA transcription translation video into YouTube. You'll find a couple really good ones that go over maybe in a little too much depth how they work, but it's important to understand all this, have a good concept, have a good grasp of these interrelations. But if there's some core things, it's really DNA to RNA to protein. Know that sequence. Know that's how it typically works. Know that when you have mutations at the DNA level, it's a big problem. And if you have mutations at that RNA level, much less so, incredibly less so. And then when it comes to us, very complicated machinery going on to regulate bacteria, it's a lot simpler machinery to regulate those genes, and we can use that very efficiently to produce things like drugs, to produce things like life-saving therapies and enzyme treatments. So that's all very, very cool, and we're going to actually get into that more in depth in lesson number 11. So I hope this has piqued your interest in that area, and then we'll go over it in a lot more depth with a lot more examples in lesson 11. Thanks so much. Have a great week, and I will see you in the next lesson.